Hey, good stuff. There's 20 people here. Now we're only missing 5. Only missing 20% of class. That's good. Okay, so this week we're leaving phylogenetics and we are going to go into mixing, which is having chairs in front here anymore. We're going to open this up, and actually as I started putting this together, I was like, I didn't really know why I started with transcriptomics and the whole genomics section. I'm not exactly sure, but here we are. We're going to start with transcriptomics. And the first thing that you should do is remember to start the video for me. Thank you. So let's talk a little bit about what transcriptomics is and why it's useful, what we're doing about it. And the transcriptome, just to kind of remind you of things I imagine that you learned in general biology, this is just the complete set of all the messenger RNAs that are in a cell. So like anytime we have an omics in front of or behind something, that's behind, right? Yeah, omics at the end. So kind of as a side commentary, about 10 years ago, maybe a little bit more than that, people decided they wanted to like stick omics on the end of everything. And some of those stuck around and some of those didn't. So transcriptomics was like one of those, and this is one of the ones that actually stuck. Funny thing, if you actually go like searching around, there's plenty of commentary out there by scientists saying that I don't know about this whole transcriptomics thing. This sounds like just another term, and then that people are proliferating all these omics, and this one actually stuck around. But there's a lot of other ones that didn't. Like the ones that really stuck around, there's genomics, transcriptomics, proteomics, metabolomics, and then past that, it really kind of, there's lots of less used terms, and they all get more and more like strange until you get like, I'm trying to think of one that was really weird, and I can't come up with one right now because it is only 8:13 or whatever, so 8:12, even earlier than I thought. Okay. Anyway, so transcriptomics is the complete set of messenger RNAs in the entire cell, and kind of why you might even care about this in the first place is if you remember, I hope you do by this point in your career, that DNA makes mRNAs, and then mRNAs are what encode for proteins. They go on to like be attached to ribosomes, and then ribosomes spit out protein, and then this, through a couple other intermediate steps in here, is what comes up with the phenotype. Now, that being said, this isn't the only RNAs being made. We're going to talk more about that in a little bit. There's a lot of other things being made as well, other types of mRNAs or other types of RNAs. But really, this is one thing that's known as like the central dogma of. Oh, this is kind of a funny side story. So, the central dogma of biology, or the central dogma of molecular biology, is kind of a funny name. Do you know who came up with that name for that? Was that?
Wasn't it Crick for once?
It was Crick. So yeah, it was Crick, and he said this, and actually, like people have asked him, like, why in the world did you call this a central dogma? Actually, turns out he didn't quite know what dogma meant, which I think is kind of funny. He thought so he thought that dogma was any idea that wasn't supported by evidence. It's just some idea, like, and he didn't want to use hypothesis over because he already named something the sequence hypothesis, in that proteins are encoded for the sequence in the DNA is actually what's encoding, and thus the sequence of the RNA is what's making encoding for the sequence of the protein, build a sequence hypothesis. And he didn't want to reuse hypothesis, but he wanted another word for something that was like an untested, unproven idea with no evidence behind it. So he's like, oh, dogma, like in religion is completely unfounded stuff, so I will use dogma as the word. Actually, it kind of means something different. Anyways, so I thought that was kind of funny. So the transcriptome then just kind of refers to the entire set of just the mRNAs that are in a cell. Now, that being said, this is the most common definition. Like a lot of things, especially like a lot of the omics that come out of this, this is not the only definition for this. Actually, I might talk about this in a minute. So then transcriptomics is just a study of the entire transcriptome. So, and let me actually come back to that. So, some people also define transcriptome as the entire set of RNAs in a cell, which is very, very different than just the whole set of mRNAs. And some people place transcriptomics as like a subdivision of genomics, which is kind of where I'm looking at here. Other people don't. The most common usage of the term is just the mRNAs. Although, just kind of be forewarned, that is not a universal definition that everyone necessarily uses. I don't know. Again, it's a recent term that kind of just evolved or popped out somewhere, and people are just kind of, yeah. But here's the thing, is like actually in the grand scheme of things, we don't really care how many mRNAs are in a cell. Like directly, that's not what's of interest. It's not like those mRNAs are directly going out and doing anything. Like, it's not like your mRNAs make your eyes blue or brown or whatever directly. What we're really interested in is in the protein expression that's happening as a result of these mRNAs. That's what we really are kind of interested in, and then thus the phenotype. Or we're kind of, another way of putting this is, we're interested in how genotype or gene expression then changes based on other circumstances, which we'll talk more about in just a little bit. Yeah, I wouldn't have had any room on that table, I guess. I would have been like this, I would have been like this, and then I would have felt really like unfulfilled in my life, and it wouldn't have been good. So then the obvious question is, if what we're really interested in is protein expression, why not just look at protein expression directly? Which would be a very good question. Which I just read something last night that you shouldn't say that was a good question after someone asked you a question in a lecture, and I thought that was stupid advice, but anyways. I know probably the person that gave that advice is going to listen to this and feel really bad about themselves, but they shouldn't. It's okay. Anyways, but the point here is that sequencing all the mRNAs is way quicker and way cheaper and more effective and easier than actually sequencing all the proteins. It's really hard. I mean, it's not insanely hard to sequence proteins, but we've gotten so good at sequencing nucleic acids that sequencing proteins is still several orders of magnitude more costly and more time-consuming than actually sequencing nucleic acids. So this is by far a easier way to go. Now that being said, people still do go out and try to quantify and sequence all the proteins that are in a cell. But that being said, that is just not something that just anyone can do. So that's difficult. See, I couldn't walk up here by standing up there. And also, I do want to talk about that there is some disconnect between mRNA expression and protein expression. It's just not one-to-one correspondence. Sometimes mRNAs can be produced and then just go on to like nothing. For instance, one thing that can happen is mRNAs can be silenced, for instance, when they bind to a microRNA that is specific for those, and then that ends up, in most cases with microRNAs, silencing that mRNA. So, just because you have mRNA present, or more mRNA present, does not necessarily mean you have more protein present. Another thing that could happen is that, so you're making protein, we're actually going through and making this, but the lifetime of that protein also plays a factor into how much of that protein that you have in the cell at any given point. For instance, if you have a very high degradation rate for a particular protein, you could have actually very low protein, or relative amount of protein, and have really high mRNA expression. And so you don't necessarily have a one-to-one correspondence here. This can happen if you have a particular protein or enzyme that breaks easily, has very few uses before it's not useful anymore, so you need to break it down and recycle it, and we have to continually be making lots more of it, or something else like this. So there has to be a little bit of caution in directly assuming that more protein or more mRNA equals more protein. It doesn't exactly mean that. So let's then talk about, well then, how do we use transcriptomics? Or how do we use this information that we would eventually get from hypothetically knowing what the complete collection is of mRNAs in the cell? And one of the really nice and interesting ways is that we can discover alternate splicing events that are happening in RNAs, or for instance, we can discover if RNA editing is happening, which is really cool. And by the way, cephalopods do tons and tons of RNA editing, which makes their genome even more crazy complex than it looks like from the genome itself. Just as a side note, really cool thing. People went looking in Antarctica at an octopus, and they were wondering, okay, so the hemocyans are affected by cold, so they must have different hemocyanin genes to actually be able to stand the cold, and then go sequence the hemocyanin genes, and they turn out to be exactly the same as octopuses from temperate waters. Not temperate, actually tropical waters. Like, really warm water octopuses have the exact same gene for hemocyanin that the cold water ones did. And they're like, this doesn't make any sense. That hemocyanin should not work in cold water. Actually, so then they started looking at the transcriptome, and they find out that, oh, it's actually being edited after transcription. It's not an alternate splicing event. It's actually direct editing. I think it's an ADU editing that's happening. Anyways, cool stuff happens. So just to give you a reminder of what these different things are, is that RNA splicing or alternate splicing is, imagine you have certain exons here for a gene. I'm going to draw it up here since that did not actually look all that good. So imagine we have these different, we have exon, we have an intron here, we have an intronic region, when we have here, and then we have another exon here, so we have C. So we have, imagine we have this gene here now with A, B, and C exons, and then these two introns. And what can happen here is we can then get an mRNA after you actually transcribe this, and then you get the RNA processing going from the pre-mRNA to the actual mRNA. You can get, for instance, this cut out, this entire thing, including B cut out, so we could have mRNA that's a C. We could have something that is a B. We could have another transcript that is BC, and we could have like a third transcript that's all of them A, B, C. So actually, if we have something like this in the genome, we could actually end up with a lot of different possible combinations of different proteins from that, based on different ways of splicing this. And this is just one kind of alternate splicing. This is kind of a variable for each of these, but there's actually lots of different ways that you can have alternate splicing happen, that you end up with lots of different proteins or different isoforms of the same protein that then have slightly different functions, or sometimes fairly wildly different functions. So that's one of the things. Discovering these alternate splicing or RNA editing events is one way that we can use transcriptomics. Also, we could look at, for instance, the effects of selection, whether that be natural selection or artificial selection, on gene expression, which is actually a really big, interesting thing. If you have anything, if you're interested in just life. Also, and here's actually the really big thing, is that we're usually generally really interested in differential gene expression between several different things. So for instance, what is the different gene expression between different species or populations within a species? What makes them different? Why are they not the same? And one of the, especially if they have like radically different phenotypes, but not very different genomes, which happens very often. How is that genome expressed differently that you actually result in these very, very different organisms? Also, we could be interested in the differential gene expression in different states of an organism. So for instance, different developmental stages are going to have vastly different gene expression. We could be interested in what is that gene expression. Different disease states, so like you have the flu or you don't have the flu. What's the differential gene expression in those situations? Also, ecological situations, and by ecological situations, I just mean in different environmental situations. Any time that you're in a different or slightly different environment. Like I take you and I put you into the snow, and you didn't want to be in the snow for whatever reason. I don't know why you wouldn't want to be in the snow, but you didn't want to be in the snow, and I just threw you into the snow, and I left you there for 2 days. Your gene expression might change. Maybe it wouldn't. I don't know. I would have to do that experiment. Do I have any volunteers? Tyler, I am taking you up on that someday. Okay. Anyways, so, and then I take out all of your mRNAs out of your entire body now. Then we can look at differential gene expression then, based on just these different situations that you put an organism into, and try to see, and this is what really interests me because I'm an ecological physiologist. I'm really interested in how, physiologically, how do organisms cope and handle the environmental situations that they're put into. And the way that you need to figure this out, well, one way that you can kind of, from a very broad brush stroke, figure this out is look at changes in the transcriptome in different situations. How are they changing their gene regulation, which then ultimately is going to affect physiology to see how do they, how do they adapt to the machines that you need to adapt them. And then also, you could be really interested in differential gene expression in different cell types. So like, for instance, different tissues. And this is actually a really big one. What makes, like a cardiomyocyte different from a liver cell? And really, I mean, this kind of ties into the developmental part of this, but what's the differential gene expression there that makes those different cells? Because they're coming from the same genome. The only difference there is how they're expressing this genome to make them into different cells. Or another one that's really big, like how do cancerous cells differ in their gene expression from normal cells? And actually this is a really big one. And actually this is a big one in which people have been throwing like tons and tons of money at, because like if anything has to do with cancer, people like to just throw money at it. I should say cancer and octopuses, but whatever. So there's actually tons and tons and tons of public information on transcriptomics in cancer cells. Like tons of it. Like if you look up anything, you can look up anything, almost. It's amazing to me. I wish there was that in other fields of study. You could do so much, but there's not. Yeah, that's cool. So, as we begin to talk about this, let's take a little look at what kinds of RNA are in a cell at any given time, because this is going to really impact how we go about this, because we're only really specifically interested in the mRNAs, but we're going to end up having to deal with all of the RNA in the cell, which is quite a bit. And it actually turns out that the vast majority of the RNA in the cell at any given time is ribosomal RNA. So that is like almost all of your RNA right now at this point in your body is ribosomal RNA. And that's about 90% of the RNA in your body, or in any given cell, is a ton. And following this up, actually, okay, we do have messenger RNAs that do create a decent portion of the remainder of the RNAome. I haven't seen that one, but apparently you can make up ohms up. Anyways, so about 1 to 4% of your entire RNAome is messenger RNA, is the transcriptome. So that's hey, there's a good chunk of it. And then we actually have a good chunk of transfer RNAs as well. If you remember, these specific ones are the ones that are kind of associated with amino acids that we're going to use to actually chunk this on the ribosome. So actually, if you think about it, almost all of the RNA in your cell by a good margin is somehow associated with the ribosome complex when it's actually doing this thing. So I guess that makes sense. But then we have tons of other RNAs that are just out there that most of them are regulatory in function, but some of them are not. And they all make up in total less than 1%, but at any given time, most of these are being turned over with a fairly high frequency. But we have small nuclear RNAs, small nucleolar RNAs, ribonuclease P, guide RNAs, microRNAs, long non-coding RNAs, Pee-wee interacting RNAs, which is my favorite. So I like how they sell Pee-wee. It is, it's like kiwi, but Pee-wee. Anyways, so we have all of these RNAs, most of which are not the RNAs that, these are not the RNAs you're looking for. I'm glad that movie never gets old. It's not like most movies I could reference from the 70s, you'd be like, what? That one? No, it's good. Okay. So let's talk about different ways. So that's kind of the background of this. Let's talk about different methods that we can use to actually quantify what kinds of things are actually there in the transcriptome. And the ones I'm going to talk about here are express sequence stacks and SAGE, which is serial analysis of gene expression, but I didn't want to write out the entire thing of serial analysis of gene expression, so I just put SAGE up there, which is what most people talk about. DNA microarrays, which is the kind of like one generation ahead of ESTs. And then finally RNA-seq, which is kind of the big dominant one at this moment. Okay, so express sequence stacks are essentially well, I'll come back to that. Well, actually, let me say it right now. The entire idea behind this is that we are just going to sequence lots of transcripts. We won't get the whole transcript, but that's okay, because you can't get the entire transcript in a single read. And so we'll just like do this lots of times, and we can kind of recreate what the entire transcript was. Or even if we don't want to reconstruct what the entire transcript was, we can identify what it was by what we did sequence. So what we do in this is we take all the mRNAs and we're going to turn them into cDNA. And we take advantage of a couple different things that the cell has made available to us. First off, reverse transcriptase, which is conveniently there from viruses that do bad things to you, but hey, we can use it as well. And then we can also take advantage is that most mRNAs have a poly A tail. So at the 3' end, we stick on, or is stuck on by the cell, many A's onto this transcript early on, so that it can identify it and use it. And we can also use that same thing. We can use this poly A tail as a primer site to start sequencing, or in this case, use with reverse transcriptase to actually turn this into cDNA, which is just the complementary DNA to the mRNAs. All that being said, just one thing to watch out here, though, for if you're thinking about doing this in sometime later today, is that not all mRNAs have a poly A tail. Most of them do. The vast majority of them do, but not all of them do. So, for instance, the mRNAs that are creating the histones, the proteins that we're winding the DNA around, do not have poly A tails. And so if you go this route, you are actually not getting like any of the histone mRNAs. So not all of them, not every single one of them do have poly A tails. So that's one of the small caveats or problems if you use a method that's targeting those poly A tails. And then what we do is we take all of those cDNAs, and then we're going to take them and clone them clone them using a cloning vector like E. coli. So essentially, just kind of a recap of what you do here. You take your cDNA or your DNA fragment of choice, and you splice this into a cloning vector. You take that, or a clone plasmid. You take that and you stick that into a coli, and let it multiply many, 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 many times, and then you get many more of your transcript. However, that being said, and then you can bust it back out of there, and then now you have many copies of it. This is actually how we used to do PCR before we knew how to do PCR. We just did cloning. Before we knew how to amplify DNA in more efficient ways. This still has advantages to doing it this way rather than just doing PCR. One of the principal ones is you don't have to know anything about the sequence first. So, for instance, you need to know something about the sequence to do PCR because you need a primer. With cloning, you don't have to know anything about the sequence at all, which makes it nice in using it for ESTs that we can do this. Like, I have no idea what's in there. Let's clone it. So, you can do that. And there's a handful of other things. People still like, for really degraded DNA, still recommend you doing cloning before you do PCR and a few other things like that. So it definitely has a place still. And then all of these, that should be a little cDNA clones. Then we start going through and sequencing those. And we're trying to get we're using some semi-high throughput stuff like pyrosequencing. So we're then busting up all these clones and then sequencing these. And each of the sequences then, because in a single read we can't get forever or really long ones like we can in Singer, we're only going to get 200 to 800 base pairs long, so not the whole transcript. You don't get the entire thing. But what we can do at that point is these ESTs then can be clustered and aligned to get the whole transcript, because again, we have fragmented these before we sequence them. All of it's there, and because we cloned it first, actually have tons and tons of copies of each transcript. So what we can do now is we can take, and we have, for instance, this, and then this, and then this, and this, and this, and kind of all the way down. And kind of how you've seen already, we can align these sorts of things, and then we can actually combine them together and figure out how they go together, each individual fragment, and get kind of an idea of what the entire thing was. Actually, it turns out this was before we launched into the Human Genome Project. This was really the only way that we had for discovering new human genes before that point, is that we were using express tag sequence or express sequence tags. Later on, it became easier once you have a genome. You don't really quite need this, but it's still useful for other reasons. It also does a really good job of being able to quantify the number of transcripts. Actually, to this day, this is still more effective at quantifying the absolute number of transcripts in a cell than things like microarrays are. Not better than RNA-seq, but better than microarrays. And this really helps if you have a genome already, because if we have a genome, we don't have to go through and align all these transcripts to each other. We can just map them onto the genome directly and figure out what they are. We don't have to do this entire reconstruction. However, all that being said, we need up to tens of thousands of reads for this to become effective at quantification of the transcripts. But we need quite a few of them. And one of those reasons that we need quite a few of them is that we're actually duplicating the transcript multiple times. Now, you need to get that to actually get good reading of this, but that also means that you need to increase the number of reads that you have to make this good and accurate. So kind of like the pros and cons of using express sequence tags. First off, this was available before a lot of other technologies were. This is one of the very first technologies that we could really begin using. In the early 90s, late 80s, we were able to use this before the genome, for other things. And it is still kind of good for gene discovery in organisms that we still don't have a genome for, which is like tons of them. We have lots of genomes, but we also have like lots of genomes we don't have. Now, the cons of this is that this is time-consuming and expensive compared to other more modern techniques. And you have some biases that are introduced by this. I've already talked about the poly-A tail and the problems with that. I mean, small problems, and if you know about them, you can deal with them, but it's still a problem. Also, there are biases that are introduced by making a cDNA library. Not every mRNA is going to respond to that exactly the same, and so you don't get necessarily a one-to-one creation with everything. So anytime that you go from mRNA to cDNA, there is a slight bias in the transcripts that you have. Now, this is a problem for all sorts of things. qPCR still uses the same thing. It goes from mRNAs to cDNAs that introduces a slight bias. Now, it's negligible, but it's still there. Okay, so that is express sequence tags. Now we're going to talk about SAGE, or serial analysis of gene expression. This is very similar to express sequence tags, but hey, we figured out at some point, especially after we had a genome, we don't need to know the entire sequence of a transcript to identify what it is. Does not matter. We just need enough of it to identify what transcript is there if we know what the entire genome is, and then be able to quantify that. So what we do here is, this is only used for quantification. We only use SAGE to actually quantify what's in the transcriptome. And we only need a short stretch of the transcript to be able to do that. So for instance, we only need somewhere around in the neighborhood of 15 base pairs to really be able to identify a transcript. Now, one of the things here is that we need a well-annotated genome. Annotated meaning that we have the entire sequence of the genome, and annotated means that we've ascribed gene names and functions to that entire genome, or a good chunk of that genome. We need to have that in order to be able to pull this off, because if we want to figure out what this 15 base pair stretch is doing, or where it came from, we need to have the genome to map that onto and figure out where this come from. So really the steps here are the same thing. First off, we're making a cDNA library like we did before with the ESTs. But at that point, the cDNAs are going to be chopped by a restriction enzyme, which is going to, on average, chop things to about 250 base pairs. These are going to then be, well, in the course here, these are going to be actually immobilized onto beads. So these are now stuck onto beads. They also have an anchoring, like an anchoring template there as well that has a known sequence. And then, like 15 base pairs into this known sequence, there is a known sequence there that we know will be trimmed by a restriction enzyme that will trim both that one and our transcript, so it cuts it at 15 base pairs. So we've now got just a transcript that has 15 base pairs on it. Then we take all of these, and we can like take tons and tons of 15 base pair sequences, and actually we put a tag on them so we know the difference between them, and then we stick them, we ligate them all together. So now we have this long stretch of DNA that's just 15 base pair stretch of transcript tag, 15 base pair stretch of transcript tag, and then we can do all of that in one run. So we can't do the entire transcriptome in one sequencing run, but in each sequencing read, I guess I should say, we can actually do lots of different transcripts. So again, we can go up 200 to 800 base pairs, each of those being like tons of 15 base pair segments of different transcripts. And so we can sequence that entire thing out and get quite a few different transcripts read or sequenced in one single pass of a sequencing reaction. Now, the pros of this is way more efficient than express sequence tags. Because with express sequence tags, actually, we end up sequencing like the entire transcript, but like several times the entire transcript, was really good if we don't know anything about the transcript or the genome. In this case, all we need now is like 15 base pairs of that, and we know where it came from with high accuracy. But we still need to get like 10,000 to 50,000 reads to actually get through the entire transcriptome, which, you know, that in reality that isn't bad with today's methods. But this can be, there should be 1,000 there. This can be covered in 300 to 1,000 reactions. Actually, about gets us that. So that's cool. That's good. So this is way fewer sequencing reactions than what we have with express sequence tags. So that improves our ability to do this quite a bit. Hey, now I take that off. What about that? Okay. Let's go through these last two just really fast. So next up, DNA microarrays. This kind of came after express sequence tags. Kind of the next easiest way to start quantifying what's happening in the transcriptome. And what happens here is that we have a collection of DNA spots on a glass slide. This is actually somewhat similar to what we saw with Illumina sequencing before, where we have all these like a lawn of oligos. Well, in this case, it's not just like general that they're all the same, and then we have stuck a adapter onto this DNA. It's not like that. It's actually like these are specific probes or specific sequences that are specific to a certain RNA transcript. And what happens then is that we wash then our transcriptome across this and allow the transcripts then to hybridize with the specific DNA spots that have the specific probes for that DNA sequence. And so, wow, that's horrible. But essentially what you have here, if I can illustrate, I can talk about this to you. That makes me really like worried about my next slide here. Okay. So you have a stretch here, and actually on here we have taken and attached a fluorescent probe onto the all of our RNA transcripts. They then hybridize then to here's our glass plate, here's our probe on here. They're going to hybridize to that, and when they do, this fluorescent probe is going to be activated, and then we can see when we actually read this, the fluorescence that's occurring at that spot. And more fluorescence equals more binding to that DNA spot, so we can somewhat quantify how much is on there. So really, the point here is that we're using hybridization rather than sequencing to do this. We have a known set of transcripts on this microarray, and we're going to allow our DNA transcripts from the transcriptome to hybridize with that. But the thing here again is, just like with SAGE, we either need to know what the sequences are beforehand to actually be able to make these DNA spots. So we have to have that genome in hand, or in another case, we need to have the EST sequences that we can create probes from. So you don't actually have to have the entire well-annotated genome in hand, but you do need to have a big enough EST library that we know enough about the sequences of transcripts that we can expect to be able to make a fairly robust microarray. Will this come out? No, that looks horrible. Let me describe to you what happens. Essentially, we make the cDNA library. We do that again. Next thing that we do is we actually couple this with then our fluorescent probes that will either be activated or deactivated when it binds to the DNA microarray. We then hybridize it with the microarray, so we're actually, those transcripts are, the cDNA transcripts are then binding to the spots on the microarray, and then we're going to get some fluorescence, and we're going to wash away everything else. And then we stick it into a machine that is represented by this thing you can't see right here. And we are going to then scan each of those dots using a laser, and then we can get the intensity ratios of all of these. Now, that's important. We're just looking at the intensity of fluorescence. We're not like counting actual sequence transcripts here. So, looking at this, first off, this is very quick and cost effective. This is still the quickest and most cost effective way to look at the transcriptome of an organism. There's no quicker and easier way to do it. That being said, first off, you have to have good knowledge of the sequences beforehand. If you're doing this on like a non-model organism, if you're not doing this on like humans, Drosophila, or C. elegans, or Arabidopsis, or some other major ones, it's gonna be really hard. But if you're like working with humans or like C. elegans, man, you can just buy these. They're also called chips. You can buy these microarrays just online, like easy. If you're working with like octopuses, you're just like, don't even try. It doesn't have it. The other thing is this isn't going to give you absolute expression. We're not counting up actual sequence transcripts. We're counting up the fluorescence, and we're going to be comparing that fluorescence then to something else of known brightness for a known amount or relatively well-known amount. So in this way, it's kind of like qPCR again, in that with qPCR, you're not actually, you're looking at this oftentimes. You can do this in ways that actually give you absolute quantification, but the quicker and easier way to do this is having housekeeping genes that you know are in relatively the same amount all the time, and you're looking at the relative fluorescence compared to those housekeeping genes. It's kind of the same here with DNA microarrays. With that, and also that kind of makes then your interpretation, your statistical interpretation of DNA microarrays somewhat foggy and difficult. And we may come back to that as we go into the processing of this data if I decide to give you DNA microarray stuff. All that being said, the usage of DNA microarrays is kind of like going out and down, even though it's quick and cost effective. The next thing that we're going to talk about is becoming more quick and cost effective. I'm not going to talk about this today. We're going to launch into this on Wednesday, but RNA-seq, which is just high throughput sequencing of the entire transcriptome, generally through Illumina sequencing, is the thing that is becoming more predominant, especially because it's becoming cheaper all the time to do high throughput sequencing. this ends up being the entire thing. Oh, and the other thing is, if you go back to DNA microarrays, they don't necessarily have the entire transcriptome as spots on there. Some of them have close to it, but no, not all of them necessarily do. So you have you kind of have problems there that is really, there's no way that you can discover like new alternate splicing events, something else like that with DNA microarrays. It doesn't do that. There's no way that you can discover like crazy RNA editing or other weird stuff that's happening will never be found with DNA microarrays, because essentially you're only going to find what you go looking for. You won't find anything new. With everything else, all the sequencing methods that we've talked about so far, well, I shouldn't say all the sequencing, not with SAGE, because again, you only go looking for what you can map to a genome, or you only find what you can map to a genome. But with ESTs and RNA-seq, you can find things that you aren't necessarily looking for, which kind of makes them more powerful in a lot of ways. And especially the more and more that we know about how fluid and kind of crazy the transcriptome is, and how alternate splicing and these other ways of subtly modifying the transcriptome has a huge impact on gene expression and phenotype. All of that variation isn't captured in DNA microarrays, and so yeah, they're kind of going out of usage. They still have, again, they're still the most cost-effective and quickest way to get any sort of quantification of the transcriptome. But understanding those limitations, they still definitely are used a good amount. Just it used to be, you go back 10 years, DNA microarrays were like everything that was used for transcriptomics, with the expression of ESTs. But like that was a dominant way of looking at this, and it's not that case anymore today. So yes, on Wednesday we'll start talking about RNA-seq, and then maybe get into actually some of the practice of dealing with transcriptomic data. But with that, enjoy your Monday, survive it, do well, and I will talk to you soon, probably.
What are they doing this week?
Not sure yet. I have two different things I'm going to